Now, let's delve into the Hardy-Weinberg equilibrium, abbreviated HWE. This is a fundamental concept in population genetics. The Hardy-Weinberg equilibrium describes the genetic expectations of an idealized, non-evolving population. To understand Hardy-Weinberg equilibrium, we need to consider 5 assumptions. 1. No selection. 2. No mutation. 3. No gene flow, meaning no migration. 4. An infinitely large population size. And 5. Random mating. These assumptions create a baseline scenario in which we can detect deviations and understand the factors that drive evolutionary change. These assumptions are also what make Hardy-Weinberg equilibrium the null hypothesis to evolution. When a population is in Hardy-Weinberg equilibrium for a gene, it is not evolving, and allele frequencies will stay the same across generations. If the 5 Hardy-Weinberg equilibrium assumptions are not met for a gene, the population may evolve for that gene, meaning the gene's allele frequency may change over time. Let's put our knowledge to the test with another activity. I'll provide you with a hypothetical population and its allele frequencies. Based on these frequencies, we can predict the genotype frequencies or genotypic frequencies in the population using Hardy-Weinberg equations. To do this, though, we must first break down the equations used for calculating Hardy-Weinberg equilibrium, right? These equations allow us to predict genotypic frequencies in a population with 2 alleles. The first equation is p squared plus 2pq plus q squared equals 1, where p and q represent the frequencies of 2 alleles. The second equation is p plus q equals 1, because the frequency of the 2 alleles added together must always equal 1, right? All parts together must equal the whole, which is 1. Let's revisit our rabbit example to help us understand. Imagine we have a population of rabbits, and we're studying the gene for whisker length. The long length allele, with a capital L, has a frequency of 0.6, and the short length allele, little l, has a frequency of 0.4. Let's assume that L is the dominant allele, while little l is recessive. Now, let's calculate the genotypic frequencies using Hardy-Weinberg equations, all right? So what you were provided with were allele frequencies. Now we'll calculate genotypic frequencies. Feel free to pause before moving on. Wonderful work! The predicted genotypic frequencies are big L equals 0.36, big L is equal to 0.48, and little l is equal to 0.16. So, those are the genotypic frequencies. But how many rabbits in the population actually have each of these genotypes? We can use our calculated genotypic frequencies here to determine the expected number of rabbits with each genotype in the population. We were told earlier that the population size is 100 rabbits. If 36% of the population has the genotype big L, that means that 36 rabbits have this genotype. If 48% have the genotype big L, then 48 individuals have this genotype. This means that we would expect 84 rabbits to have long whiskers, right? Similarly, if 16% of the rabbits have the genotype little l, then 16 rabbits have this genotype. This means we would expect only 16 rabbits to have short whiskers in this population. Instead of having you try your hand at these calculations here, you're actually going to do so in one of your upcoming activities. So feel free to review this material before moving on to that activity. Sound good? Now, that's how we use Hardy-Weinberg equilibrium to calculate allele and genotypic frequencies. But these calculations serve as quote-unquote ideal values. Remember, Hardy-Weinberg equilibrium is essentially the null hypothesis to evolution. So, if the calculated values here differ significantly from those we see in reality, then we would reject this null hypothesis and say that the population is likely evolving, right? This means that deviations from predicted allele frequencies, calculated using Hardy-Weinberg equations, can provide us with insights into evolutionary processes at work. If the observed frequencies differ significantly from the predicted calculated frequencies, that means what we are observing is very different from Hardy-Weinberg equilibrium, right? Suggesting that evolutionary forces are likely influencing the population. So to clarify, I'm using terms like expected versus observed. So expected or predicted values are those we calculate using Hardy-Weinberg equations, while actual or observed values are those we actually see and count in real populations. So in our rabbit example, if our expected values were 84 rabbits with long whiskers and 16 rabbits with short whiskers, but when we went out to our population of 100 rabbits and counted up each rabbit in the actual population that have long or short whiskers, and the number we observed were significantly different from 84 and 16, then we could say with confidence that evolutionary forces are likely influencing this population of rabbits. Can you think of any examples of how deviations from predicted allele frequencies can occur, and what they might mean in evolutionary terms? Deviations from predicted allele frequencies can occur due to a variety of factors, such as natural selection, mutation, migration or gene flow, a small population size or genetic drift, and non-random mating. These deviations indicate that evolutionary processes are acting upon the population, leading to the changes in allele frequencies over time. Well, congratulations! You've explored the evidence for evolution and the dynamics of populations. We covered the definition of species and populations, the role of alleles and mutations in genetic variation, the concept of Hardy-Weinberg equilibrium, and how to calculate genotypic frequencies. We also discussed the evolutionary implications of deviations from predicted allele frequencies. Keep delving into these concepts and just marvel at the intricate mechanisms that drive evolution. Evolutionary biology continues to unlock the mysteries of life's diversity. Keep questioning, exploring, and embracing the wonder of biology. Till next time.